Good day. Today we're going to talk about mitosis. But first, what is mitosis? Mitosis is the nuclear division that includes cytokinesis and produces two daughter cells at the end of the whole process. Follow me. First off, let's list down the phases of mitosis, which are interphase, prophase, metaphase, anaphase, and last, telophase. Interphase, first we're going to talk about interphase, which isn't really part of the processes, but it's still part of mitosis. Now, version 1 of this, put it simply, interphase is the little rest stop for the cell between cell divisions. During this phase, the cell creates the various materials it will be needing for the next cell division, such as protein, organelles, and molecules, which the cell will use later on. Version 2 of this, version 2 of interphase, is divided into 3 stages of the cell cycle, which is S, G1, and G2. There's also G0, but we won't be discussing that in this video. In the G1 stage, which takes place between the beginnings of mitosis and the S stage, is where the non-reproductive functions of cells start working, such as increases in growth, creation, and size of organelles, and increased enzymes activity for also later use. The S stage is characterized by DNA replication, is where the cell creates more materials like proteins, which is also found in the other one. The stage that follows after that is the G2 stage. This is the shortest of all stages because it begins after the DNA replication from earlier it finishes. During this stage, most of the organelles and molecules for cell division are produced. Also, the materials that are needed for the movement of chromosomes and cell replication gets organized. Moving on to the next phase. This would be prophase. Prophase is the longest lasting phase during the whole process of mitosis. It lasts around 50% of the whole thing. During this phase, the chromatin in the nucleus starts to condense into chromosomes, which can be seen on a microscope. In this video, we're going to be using coins for the chromosomes. Peso coins, if anybody's asking. Give me a moment. Also, during this phase, the nuclear envelope disappears and the centrioles separate and move to opposite ends of the cell, which should end up serving as the base for the spindle fibers, which aren't really present right now, but will be. Next one is metaphase. Metaphase is the shortest phase, only lasting a few minutes. The split centrioles, which are now spindle fibers, arrange the chromosomes into a line. So it looks like this. These are 2 separate chromosomes. Then they get lined up in the middle. This is the metaphase stage. and they're all connected as well. In the middle of the cell, this line is referred as the metaphase plate. The purpose of this line is to ensure that during the next phase, when each chromosome is separated, each new nucleus gets an even share of the chromosomes. Next up is anaphase. The aligned chromosomes from earlier get split evenly and moved to the opposite sides of the cell. Now, for now, don't pay attention to the metaphase and the prophase, and the metaphase plate words. This is anaphase, where they get split into two. These are spindle fibers, as I said I would put on before. So as you can see, the two chromosomes which were formed from before, they were split into their halves and divided equally. And now for the second to the last phase, telophase. During telophase, the chromatids move to the opposite sides of the cell, joining the dispersing chromosomes. So the spindle fibers disperse as well, and the cytokinesis can also begin during this stage. And last but not the least, cytokinesis. For animal cells, a fiber ring made up of proteins called actin start pinching the cell until it breaks into two daughter cells, nucleus and all. For plant cells, a wall called the cell wall is synthesized between the daughter cells. So it ends up looking a bit like this. Cell 1, cell 2. And that is the process we call mitosis. Thank you.